So good evening students, कैसे हैं आप सब how are you all doing? All doing good? So soon we start in 5 minutes. Today we will deal with the part of mutations, cytoplasmic inheritance with the plastid and mitochondrial DNA type of inheritance, and things that induce mutations, and a little bit about of CIB techniques. So we will be conducting major four topics today. So slowly and slowly we go. Let's see how far we would go. So just let's wait for another 5 minutes, then we start. Let all other students join us also. as the time passes, it's 8 now. I think we could start now. Uh, so let's start first with the cytoplasmic inheritance. Basically, students, we discuss in this topic that there is an inheritance that is happening because of the uh, egg and sperm, like stamen and pollens. But sometimes there is a inheritance is happening just because of the cytoplasmic DNA material, and it's also happening because of mainly the female characteristics and not because of the male. So all the inheritance part that is present in the cytoplasmic material, whether it's a mitochondrial, plastid, or chloroplast, anything is present in our DNA material, and that is being inherited to the next generation. That is basically is your cytoplasmic inheritance, and our topic will go around the same topic today. So this is from your unit 1. We are done with the 4 topics in 2 days, that is history, Mendel's law, type of gene action, qualitative and quantitative traits, multiple factor hypothesis, and today we will be dealing with the cytoplasmic inheritance and mutations. We will deal with the mutations also, and how they are linked to the sex-linked autosomal mutations and CIB technique. So we did do our part till here today. So let's start with the cytoplasmic inheritance. So inheritance of the most of the characteristic in eukaryotic organisms shows the following features. So there are two kind of features. One is that their contribution of both this contribution by both male and female parents are equal, so that the results are reciprocal, crosses are identical. Reciprocal crosses, what are reciprocal crosses? They're the crosses which are done back with the parents. So in the contribution of male and female parents are equal, so the results in the reciprocal crosses are equal because both of the gametes are being transferred. And segregation produces a characteristics ratio of 3 is to 1 whenever we do the back cross with the or reciprocal cross, and after generation is the monohybrid cross is typical of 9 is to 3 is to 3 1. So on the dihybrid cross. So these features of inheritance were first demonstrated by Mendelian, as we have discussed in previous lectures, and this inheritance pattern is called Mendelian inheritance. And it has been universally accepted that these inheritance that we are talking about, they were all happening because of the chromosomes that are present in the eukaryotic nuclei, and these are sufficient evidence that these chromosomes are playing in that role, and such genes that are present in the chromosome are called nuclear genes, or more commonly as called as genes only. So that was something about in normal inheritance that is happening according to Mendelian law because of the chromosomal genes present in the chromosomes. Chromosomal because of the chromosomes having a nuclear genes or genes present in it. But there are some organisms which were not showing a Mendelian inheritance; they were showing non-Mendelian inheritance pattern. So what was observed in those cases? It was a consistent difference between the results from the reciprocal cross. So whenever they do the reciprocal cross, they had a different progeny, and mostly that is the trait that is been transferred. They were mainly from the female parents, not from the male parents. And afterwards, if we see this 9 is to 3 is to 3 is to 3 1, 9 is to 3 is to 3 is to 1 in dihybrid crosses in F2 generation, but we don't see any segregation in the F2 generation and also in further generations. So everything remains same as per the whatever the female parents are, it remains the same. So characters showing non-Mendelian inheritance may be grouped under three categories. So the reason behind it could be these are related to the cellular structures and patterns, intracellular parasites, symbionts and viruses, or these are associated with the DNA containing cell organelles such as mitochondria and chloroplast. So these might be happening also because of the DNA that was present in the in your cytoplasm, and it is done because of the mitochondrial and chloroplast DNA. So in the non-Mendelian inheritance, there is also by evidence. The first evidence of cytoplasmic inheritance was given by the Correns in the Mirabilis yellapa plant and by Bauer in the Pellegrinium zonale in 1908. So, in the case of cytoplasmic inheritance, generally the character of one of the two parents, usually the female parents, is transmitted to the progeny. That's the main thing that is happening in this case. And whenever we do the reciprocal cross exhibit differences for such character, we don't see segregation. There's a lack of segregation in the F2 generation and also in further generation. So such generation, such inheritance in which we don't see any differences in the F2 generations, we called it as extranuclear inheritance, extra chromosomal inheritance, or maternal inheritance. Remember these terms, students. So these are new terms are coming for you in the genetics part. So like we are touching the advanced genetics part now. So something in the cytoplasmic inheritance, whenever there is no subsequent changes in the in the F2 generation, uh, so that generation, that inheritance is referred to as extranuclear inheritance, extra chromosomal inheritance, and maternal inheritance. So genes governing the traits showing cytoplasmic inheritance are located outside the nucleus. So the genes that were dealing with the cytoplasmic inheritance, of course, they are present outside the nucleus and in the cytoplasm. Hence, these genes are referred to as plasma genes. They have different names: cytoplasmic genes, cytogenes, extranuclear genes, or extra chromosomal genes. So these are the various names to the genes that are present outside your cytoplasm, that is plasma, cytoplasmic, cytogenes, extranuclear genes, or extra chromosomal genes. And the sum total of all genes present in the cytoplasm of cell is known as plasmon, whereas the genes present in the plastid, it is known as plastron. So sum total genes present in the cytoplasm itself, it is known as plasmon, and ones that are present in only in the plastid, that constitute your plastron. So some few characteristics of cytoplasmic inheritance. So reciprocal differences. Reciprocal cross shows marked differences for the characteristics governed by the plasma genes. In most cases, plasma genes from only one parent, female parents are transmitted, and phenomena is known as uniparental inheritance. So as the female is the only one that is transmitting the plasma genes from one generation to another, and this phenomena is known as uniparental inheritance. And there is a lag of segregation. So in F2, F3 subsequent do not show segregations for cytoplasmic inherited traits. This is because F1 individual generally receive plasma genes from one parent only, because they are receiving from the one parent. So we don't see much changes in the F2 and F3 generation. There is irregular segregation in the biparental inheritance. That is, in some cases, sorry students. Uh, Irregular, in some cases, plasma genes from the both parents are transmitted, and then it is known as biparental inheritance. And further, somatic segregation, plasma gene generally shows somatic segregations during mitosis and feature of rare occurrence in the case of nuclear genes. So here, instead of your zygotic segregation, somatic segregation is happening. and this association with the organelle DNA, that is, several plasma genes have been shown to be associated with cytoplasmic DNA or mitochondrial DNA. Uh, Further, taking to the characteristics of cytoplasmic inheritance, nuclear transplantation. So, if nuclear transplantation revealed a trait to be governed by genotype of cytoplasm and not by the nucleus, or cytoplasmic inheritance of the trait is strongly indicated. So, whenever the genotype of cytoplasm is being shown instead of the nucleus, cytoplasmic inheritance is governed to be governed by cytoplasmic inheritance. So in nuclear transplantation, mainly nucleus of the cell is removed and replaced by the nucleus of another genotype from a different cell. And generally, nuclei of somatic cells are transplanted into zygotes before the first mitotic division is initiated. Further, in this case, there is a transfer of nuclear gene through the back crosses are happening, and last not the least, mutagenesis. So there are some mutagens such as ethidium bromide are highly affected by mutagens, leading to the nuclear genes, whereas your nuclear genes are not affected, but your plasma genes are highly affected because of them. And there is a lack of chromosomal locations in many organisms. Extensive linkage maps of nuclear genes are available. If a gene is shown to be located in one of these linkage groups, it cannot be a plasma gene. Failure to demonstrate that location is in one linkage group of organism is indicated of cytoplasmic location, but this is highly tentative in that case. And there's a lack of association with the parasites, symbionts, and viruses. In many cases, cytoplasmic inherited characters are associated with the parasites, symbiote, or viruses present in the cytoplasm of the organisms. Such cases cannot be regarded as a case of cytoplasmic inheritance. Only those which are not associated with with the parasites, symbionts, or viruses can be governed by the plasma genes. So, in nutshell, so the cytoplasmic inheritance which are not associated with the parasites, only they are could be governed by the plasma genes. Otherwise, if they are governed by these viruses or parasites, then there is no concern with the cytoplasmic inheritance in that case. So further, uh, let's take it. Uh, just a second. Yeah, so this is now about true cytoplasmic inheritance that is governed by the chloroplast or mitochondrial traits are associated with the DNA. And they are also offered to as organelle inheritance, plastid inheritance, and mitochondrial inheritance. So let's start with the first plastid inheritance. So inheritance which is governed by the plastid characteristics in the plants due to the plasma genes located in the plastid is known as plastid inheritance. So it's same like cytoplasmic inheritance. Instead of in the cytoplasm here, the genes are present in your plastid, and that is known as your plastid inheritance. It was first case of cytoplasmic inheritance discovered by Correns and Bauer in 1908. So what they discovered is variegation. What is variegation is the spot color of white and yellow. White and yellow spots of variable size on the green background of leaves. There's a green background of leaves. So here you see white and some yellow spots on the leaves, and this may be produced by the environmental factors or some in nuclear genes or some in cases plasma genes. So all could be the reason behind it of producing it. And inheritance of plastids in mirabilis and jalapa. The inheritance of plastids in 4 o'clock mirabilis jalapa was first discovered by Correns. So what they did is they took branches having normal leaves, normal green leaves of the same plant, while other branches they took one from the pale green, white leaves, and also some having variegated leaves. So this is how the table they have generated. So they have a type of branch from which flowers are chosen for the pollination. So for pollination, they choose some flowers and they choose some branch from which pollen was obtained, and then type of leaf that what they could find out grown from the seeds. Then flowers on the branch with the normal green leaves produce green seeds that grown into plants with normal green leaves, irrespective they are pollinated by from normal green vegetative or pale green leaves. So here in the first case, when the green plants are pollinated with the green or variegated or pale green, they all produced all green plants. But when the variegated green plant uh, got pollinated with the green, variegated, pale green, they have all the three types were coming up: green, variegated also, and pale also. So in all the cases, whereas in the pale green there was no variegated part was coming. It was mainly coming because green and pale green. So there was something that is variegated green, that thing was coming in all the cases. There is some reason behind it. What was the reason? It is clear that variegation is determined by the genes transmitted through the female, and it is influenced by the type of pollen used. So it is depending upon the female, and it depends upon the type of the pollen used actually. So these agencies are chloroplasts. So they are capable of self-duplication and transmitted from one generation to another generation through cytoplasm of the egg. So that's why some born on green branch have three genes only, green plastids. Seeds born on the pale green branch have three genes only, pale green plastids. Seeds born on the variegated will have green, pale, or a mixture that is variegated of the two types of plastids. So variegation is thus hereditary character determined by the self duplicating extranuclear particles called plastid. So that the thing that was we're seeing in the middle case was happening because of the plastids actually. So neither the nucleus of the female gamete nor the male gamete is involved in the control of this type of hereditary character. So no nucleus was involved, nothing female gamete, no male gametes, mainly the plastid was the reason that was leading to this plastid inheritance. This is the same example of uh, done in the maize. Uh, I will skip this. It's not so important. Main thing was this one actually, that to teach you how plastid characteristics are being used. Then, in case of male sterility, in maize, pollen grains of such male sterile are aborted, so they are not being used once the pollen grains of such male steriles are taken, and this male sterility is transmitted only through female and never by the pollen. So it was later recognized that cytoplasmic male sterility in maize results from the alteration of the hereditary units in the mitochondria. So cytoplasmic male sterility was happening mainly due to the reason behind there was alteration in the hereditary units of the mitochondrial DNA. Then to take you on the same chapter about we will talk about now paramecium, inheritance of Kappa materials, which will let us know about cytoplasmic inheritances for with further example. That is, in Paramecium aureolea, there are two kind of strains present. One is called killer, which secrete a toxic substance such as paramecium. The strain is sensitive, which is killed if it's contact with the paramecium. So there is a one killer, one is a sensitive kappa materials, and in the cytoplasm of the killer strain, the kappa materials, cytoplasmic DNA are present. So they are the cytoplasmic DNA, these kappa materials of cytoplasm, these killer strains are present, and they are absent in the sensitive strains. They are not present in the sensitive strains at all. And the transmission of kappa particle is through the cytoplasm, but the maintenance of kappa particles, production of a paramecium is controlled by the gene small k, and we assume that a killer strain carry dominant allele and sensitive one have the recessive genes small k small k. So whenever the conjugation happens, there's exchange of nuclear materials are happening. So conjugates produce small k when conjugation is for normal time. and it will also produce killer daughter cells, sensitive will produce sensitive daughter cells. So that is normal. Killer will produce killer and sensitive will produce sensitive daughter cells. So let's see on conjugations. Um, Here you can see there is a sensitive, small k, there is a killer, capital K, and this is sensitive is capital K. But here your nuclear Kappa materials are present inside this. So there's a cross done between these two. So with the cross you can see there's small k, capital K, capital K, capital K, capital K. And then when the cross is done, you have delayed separation. Separation is delayed. So you have small k, capital K, capital K, capital K, capital K, capital K, capital K. And then you have various uh, there are some immediate separations also when the immediate separation is done. So you can have capital K, capital K, small k. So some will be have paramecium or release killer cells, some will not have. In this case, in delay pattern, both have the paramecium killer cells, um, and in this case also, and with the immediate separation in this is right cases also. So that's how your killer strain is being transferred because of the cytoplasmic inheritance. And also this inheritance could be done via mitochondria. So we have discussed about plastid and also nuclear and also paramecium killer strains. Now comes the third part, inheritance through the uh, mitochondria. So mitochondria can self-replicate and represent another system, another genetic system in the cell. Of course, the amount of mitochondrial DNA is so small that representing cell of that 1% nuclear DNA is mammalian cells, and it can code for a part of protein in the mitochondria. The synthesis of cytochrome found in mitochondria, for example, is known to be present in minute amount in cytoplasm under the control of nuclear genes. Therefore, it is suggested that both mitochondria and chloroplast seems to have semi-autonomous existence, and their DNA forms the basis of genetic systems and separate from that in the nucleus. Then there is episomes in bacteria. So hereditary particles can be found in two states, either in the autonomous state in the cytoplasm or in the replicate independently of the chromosome, or in the integrated state incorporated into the chromosome. Particle with such properties is known as episomes and includes such things factor called sex factors. Episomes are apparently not essential to the life of the bacteria because they or may not be present. If they are absent, they may acquire only external source. In bacteria, E. coli, sex is determined by the presence of the absence of these sex factors F. And male bacterial have the sex factor, and this factor is responsible for the transfer of DNA from male to female bacterial part. And these sex factors are cytoplasmic particles actually. So what is the significance of cytoplasmic inheritance? There could be mutation of chloroplast DNA and mitochondrial DNA leads to generation of new variations. It could also manifest heterosis in the case of mitochondria and cytoplasmic male sterility. Several crop plants like maize, pearl, millet, sorghum, cotton, etc. could be produced in that case. So that's it for this slide. So students, just take 5 minutes break. We meet us at 8:30, and then we starts with the another part of mutation. Then okay, we just meet in 5 minutes. Class, I'm back. Let's start with the mutation, the next topic in your chapter in the Unit 2, Unit 1, that is mutations and methods of inducing mutations. So let's start. So anyone could tell me what is mutation is? What is mutation? Okay. Uh, Most of you are sleeping, I guess. Is a change? Yes, somebody came. Alteration of the genetic sequence. Yes. There's alteration of expression. Yes, true, true. Change in the DNA sequence. So all these belongs to your mutations happening at your DNA level. So nature's intention is that the exact genetic information from both parents will be seen in the offspring, and uh, and that and then the critical stages of fertilizations. However, it is possible for the genetic information to mutate, which in most cases can result in the fatal or negative consequences in the outcome of the new organism. So, one well-known example of mutation is non-disjunction. In non-disjunction mutation, what happening is the spindle fibers fail to separate during the meiosis, resulting in gametes with one extra chromosome and other gamete lacking the chromosome. So there is one extra chromosome and other side gametes lacking that extra chromosome. So in this non-disjunction occurs in chromosome number 21 of human egg cells, leading to the Down syndrome in humans. And this is happening because of the 47 chromosomes are present, as the normals are the complementary 46, and this is happening because of the non-disjunction, and that's quite dealing with your Down syndrome disease also. Fundamental structure of chromosomes subject to mutate, which mostly likely to occur during the crossing over at meiosis, and there are a number of ways in which the chromosome structure can change, which will determinantly change the genotype and phenotype of the organisms. So, if the chromosome mutations affect the essential part of DNA, it is possible that mutation will abort the offspring and because before it has a chance of being born. So whenever there is some mutation at chromosomal part is affecting and which is affecting the essential part of DNA, and this will abort the offspring coming up, and it will chance that before a newborn is born. So it will stop within before the pregnancy would happen. So mutation, most organisms is happening has segments of DNA molecules, and mutation refers to the heritable changes happening in the genome, excluding the results from the incorporating genetic material from other organisms. So mutation is abrupt qualitative or quantitative changes in the genetic material of the organisms, and this could be intragenic and intergenic. What is intragenic? Now we will discuss various types of mutations. So first comes the intragenic and intergenic mutations. So intragenic or point mutation includes alteration in the structure of DNA molecule within that gene. So within the gene, this is happening of your molecule. And in this, there is a change in the normal base sequence of the DNA molecule. So there is a point mutation, normal base sequence. So like from A to T or T to G, G to C, like this change is happening. And this change results in the modification of structural characteristics or enzymatic capacities of the individual. Once your DNA is changed, then it will lead to the structural as well as enzymatic characteristics of the individual change in them. And unit of gene mutation is known as muton, and it may consist of one or many nucleotide pairs. So one or many could be done within the intragenic one. Whereas the intergenic is of which chromosomal changes in structures are happening, involve long regions of DNA. That is, many genes are involved in the uh, chromosomal changes. So these includes deletion or addition of segment of chromosome. So one segment of your chromosome is deleted or added, resulting in the deficiency or duplication respectively. Either duplicated or it make a deficiency of your genes in that case. So in large deletions, a base sequence corresponding to the entire polypeptide chain could be lost sometimes. Such mutations are quite useful in the genetic mapping. Then comes the germinal and somatic mutations. So eukaryotic organisms producing two primary cells, one for the inheritance, that is germ, and the somatics for the growth of our cells. So mutation can occur in either cell types. If a gene is altered in the germ cell type, the mutation is known as a germinal mutation, because germ cells give rise to gametes, and some gametes will carry the mutations and it will be passed on to the next generations when the individual successfully mates in that case. So typically germinal mutations are not expressed in the individuals containing the mutation, whereas the somatic cells give rise to all non-germline tissues. Mutations in somatic cells are called somatic mutations, and because they do not occur in cells that give rise to gametes, mutation in this case will not pass to the next generation by sexual means. To maintain this mutation, the individual containing the mutations must be cloned, and two examples of somatic clones are navel oranges and red delicious apples. So in this case, somatic mutation will transfer to the next part, whereas the germinal will, oh, somatic will not transfer because they are not germ cells, where in the upper case the mutation will transfer because of the germinal cells. Then comes your somatic and induced mutations. In general, the appearance of new mutation is rare event, but most of the mutations that originally studied occur spontaneously, and this class of mutation is termed as spontaneous mutations. That the one mutations which occur on their own, and they are known as spontaneous mutations. And these mutations clearly represent only a small number or all possible mutations. Another side, to genetically dissect and biologically system further, new mutations were created by scientists by treating an organism with the mutagenizing agents, and these mutations are called induced mutations. The one with the done with inducing with the mutagenizing agents, these mutations are called induced mutations, and spontaneous which occur happen on their own. So spontaneous mutations rate varies. Large gene provides a large target meant to mutate more frequently, and on the other side, they are in the case of induced mutations, it could be done by several methods. It could be done with the help of radiation, chemical, or transposon insertions. The first induced mutations were created by creating a Drosophila with X-rays. In addition, X-rays other than other types of radiation treatment have chosen proven useful, including gamma rays and fast neuron bombardment. These treatment can induce point mutations or deletions also. Some other types of mutations now. Morphological mutants are the one which appears or the outward appearance for individuals, like plant height mutations, changes from tall to short one or from smooth to round seeds. Then there's a biochemical mutations which have lesions in one specific type of enzymatic pathway. For example, in bacteria, biochemical mutants need to be grown on the media supplemented with specific nutrients, and such mutants are called auxotrope. And the morphological mutants are the direct results of the mutations in the biochemical pathway. So both mutations are interrelated, the morphological and biochemical, as morphological mutants are the direct results of the mutations in the biochemical mutations, as in the change in the enzymatic pathway um, of your organism. Some more, in humans, albinism, the white skin is a result of mutations in the pathway where we convert the amino acids tyrosine into the skin pigment melanin. So, so melanin is required for our skin to be brown, but that has been convert, that has been mutated, and that amino acid is not able to mutate it anymore. Similarly, cretinism results from the tyrosine to thyroxine pathway is mutated, and that pathway is mutated. For some mutations to be expressed, the individual needs to be placed in specific environment, and this is called as restrictive conditions. But if the individuals grow in other environment, the wild type phenotype is expressed, and these are called conditional mutations. Mutations that only expressed at specific temperature, usually elevated, can be considered to be the conditional mutations. Further, there is also called lethal mutations, which are all also possible at the time of death, which could lead to the death of individuals. So that does not occur immediately. It may take several months or several years also, but it expected during this lethal mutation, person living life is reduced, and these mutations are considered to be as lethal mutations. If mutation occurs in a allele and the function of for which encode is also lost, that term is known as loss of function mutations. When the function is lost, then that term is itself is known as loss of functions mutations. And the degree to which function is lost can vary. If the function is entirely lost, the mutation is called null mutations. If it is all possibility that some functions may remain but do not at the level of wild type allele, these are called leaky mutations. So there are also some chromosome mutations are also happening, which is happening at the gross changes in the chromosome. So first is the change in the number of chromosomes. So number of chromosomes are changed, and that is known as euploidy, variation in the number sets of chromosome. So in a haploidy, one set of chromosome, that is A, B, C. In polyploidy, three or more set of chromosomes are happening. In triploidy, three sets, tetraploidy, four, pentaploidy, five, hexaploidy, six, septaploidy, seven, octaploidy, eight. Sorry, and like this, it's keep going on. Then comes the aneuploidy. In this case, variations in the number of chromosome of set. So there is a reduction in the normal number of chromosomes. If it is monosomic, that is the loss of one chromosome. That is, there is 2n minus 1. From ABC, it's become AB. If it is two times double monosomic, loss of two different chromosomes, 2n minus 1 minus 1. Then there's a loss of pair of homologous chromosomes, that's 2n minus 2. When there is an increase in number of chromosomes, then it is known as polysomy. That is, trisomy is present of one extra chromosome, 2n plus 1. Double trisomy is present of two different extra chromosome, 2n plus 2, plus 1 and plus 1. Then tetrasomy, pair of like two chromosomes extra. And pentasomy, hexasomy, septasomic, like this it goes on. Uh, there could be also changes in the structure of chromosomes, that is loss or addition of segmentation of chromosomes. There could be deletion, a loss of segments, or duplication, repetition of a segment of a chromosome. Change in the normal arrangement of genes in the chromosomes, and these could be two types: translocation and inversion. In the translocation, exchange of segments between two non-homologous chromosomes resulting in the new chromosome. An inversion is the change in the linear order of gene by rotation of a section of a chromosome through 180 degrees Celsius. Then there is a gene mutation, point mutation, which could lead to the change in the sequence of a gene by deletion, insertion, substitution, or inversion. Then comes the next term, as in your rate of mutation. Uh, so in this case, you can see that there is a frequency of spontaneous mutation is usually low. It doesn't happen at a very high range from 10 to the power 7 to 10 to the power 12 per organism. And the rate of detectable mutation is, in average gene is 1 in 106. So 1 in 106 genes, this rate of mutation is happening. So most methods of estimating the rate of mutations tend to be estimate their frequency due to many reasons. First, lethal mutation, and also secondly, the mutations which leave only a slight change in the phenotype may remain undetected. So mutations which occur frequently in certain region of the gene than in the others, the favored regions are called in that case are hotspots, where it's very common to have mutations. These regions will be called as hotspots. And the mutations involving single nucleotides can revert to normal gene structure. Most single gene mutations are reversible. In many cases, the rate of reverse mutation is similar to the rate of forward mutations. In rare location, the rate of forward mutation is much greater than the rate of the backward mutations. So, what are the effects of mutations on the phenotype? According to the effect on the phenotype, mutations may be classified as lethal, subvital, and supervital. So, lethal mutations results in the death of person in which they occur. Subvital is the one which has chances still could survive. Supervital mutation is the improvement of biological fitness under certain conditions. So there may be also mutations which are neither harmful nor beneficial to the organisms in which they are occurring. So how does this mutation acts? So as mutation in other sections, genes act by controlling the rate of productions of specific proteins. The scheme of protein synthesis in most organisms as follows. So first is gene. Your DNA produces a complementary mRNA, which has codons consisted of nucleotide triplets. Then transfer RNA molecules, each forming a complex with specific amino acids, have three free nucleotides which form the anticodon. The alignment of molecules on mRNA depend upon the complementary codon-anticodon pairing. The sequence of amino acids molecular in enzyme and hence the structure and function of enzyme depend upon the nucleotide sequence of mRNA. So this is telling about us about translation and transcription here, that during this protein synthesis that your DNA is producing mRNA and mRNA is producing anticodons, and anticodons are being coded by the codons of the tRNA, and hence an amino acids are produced via tRNA with the help of ribosomes in the case of uh, translation. So in that case, nucleotide sequence in DNA, it will be seen in that change in the sequence of nucleotides of DNA will result in corresponding change in the nucleotide sequence of mRNA. This may result in the alignment of different tRNA molecules on mRNA. Thus, the amino acid sequence and hence the structure and properties of the enzyme formed will be changed, and this may affect the trait controlled by the enzyme. So, yeah. So next, molecular basis of mutations. So gene mutations at the molecular level involve modifications of one base by another, or addition or deletion of one or more bases. So mutations may be spontaneous or induced mutations. Mutations which occur under natural conditions, we have discussed that they're spontaneous, which are done in induced condition, they are the induced mutations. So the truly spontaneous mutations will dealt with theirs arising from the tautomerism. So what is tautomerism is? Tautomerism is the ability of molecule to exist in more than one chemical form. So all four common bases, that is adenine, guanine, cytosine, and thymine, they have unusual tautomeric forms, which are however rare to having their different chemical form of the all four bases. So normal bases of DNA usually present in the keto form. As a result, automeric rearrangement could momentarily transform into the enol form from keto to enol form, in which distribution of electrons is slightly different. Normal base pairing in DNA is A, T, and G, C, and the tautomeric forms are, however, capable of unusual base pairing like T, G, G, T, C, A, A, C. So these kind of uh, forbidden base pairing could be done also, which will lead to the misreplication of DNA, giving rise to mutants in some progeny. And thus, a rare tautomerism of adenine pairs with cytosine leads to the G pairing. This leads to GC pairing to the next generation. And In the induced mutations, that was something about spontaneous, and induced mutations could be done via chemical mutagens such as radiations like X-ray, gamma ray, and UV light, or it could be done using the help of chemical mutagens like mustard gas was first chemical mutagen discovered in 1950, and we can classify them according to their uh, the way they are working out. So first is base analogs, which get incorporated into DNA, and they're one which are modifying the purines and pyrimidines and agents labeling bases, and agents producing distortion in the DNA. So agents including 1 and 3 requires replication of their action, where the second one can modify non-replicating DNA. We will deal about them in quite details in our next chapter with the one which are doing induced mutations. Uh, they are the one they're producing point mutations, chemical mutations. With the chemical mutagens, there are two classes basically, alkylating and base analogs, and each has specific effect on the DNA. Alkylating agent is the one such as ethyl methane sulfonate (EMS), ethyl ethane sulfonate (ES), or mustard gas that can mutate both replicating and non-replicating DNA. So by contrast, base analog only mutate DNA when the analog is incorporated into the replicating DNA. So each class of chemical mutagens has specific effects that can lead to the transition, transversions, or deletions also. Scientists now are using power of transposable elements to create new mutations. So they are the mobile pieces of DNA that can move from one location to another. In the case of genetics, often when they move to new location, the result is new mutant. So that's how the transposable elements are important. So it was a quite deep and quite big lecture for all of you. I hope I went through slow pace in order to let you understand more about various kind of mutations, how things are happening, how mutations is happening, and many different parts. So on this point, let's have a 5 minutes break again, and then we start with methods of inducing mutations. But yeah, transpose insertion is a whole new chapter, so I just want to touch a small part to you. It's a full in the MBBT we do that part in the molecular biology techniques. So where we add maybe you can ask me personally. I can explain you better, but here it's not possible in these lectures. Do presence of brown parts or spots on skin, also called albinism for white people? No, 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 no. Albinism itself is having a white skin actually. Albinos, there's mulattoes, albinos, and having that brown parts, it's a different skin disorder, nothing to do related with albinism. It's a different stuff. Don't mix it up. Yeah. So we meet at 9 then, yeah, and then at 9 we start the last topic of your today's topic. Finish with the today's things then. Okay, I'm coming back. Let's wait for 5 minutes only. Okay, students, let's start with our end part of our today's lecture, last series, methods of inducing mutations. And this CIV technique I will deal in the last, at the end of your lectures. So that's that's something with another day. So mutations. So we have discussed about a lot of about mutations, how it is done, and now it's time to discuss about mutagens. So they are mainly of two types: physical and chemical. Physical mutagens are the one which are having different radiations. They are called physical mutagens. So it could include electromagnetic spectrum waves having a shorter wavelength and higher energy than the visible light, that is UV ray, X-ray, gamma ray, and cosmic ray, or maybe particulate radiations produced by the decay of radioisotopes. So radiations was the first mutagenic agent known. Its effects on genes were first reported in the 1920. Radiation itself was discovered in 1890 by the Roentgen discovered X-rays in 1895. Then Becquerel discovered radioactivity in 1896, and Marie and Pierre Curie discovered radioactive elements in 1898. So these discoveries others led to the birth of atomic physics and other understanding of electromagnetic radiations. Radiations are grouped into two classes depending on the kind of effects they have on the atoms in their path, that is ionizing and non-ionizing radiations. Non-ionizing radiations are including ultraviolet rays are the only non-ionizing radiations with mutagenic properties. The wavelength ranges from 100 to um, 3900 angstrom, and they are specifically absorbed by purines and pyrimidines present in our DNA. Further, the maximum absorption of UV rays by DNA as well as by pyrimidines, particularly thymine, occurs at the wavelength of 254 nanometer, which is also the most mutagenic wavelength for the UV. Mutagenic action of UV is a consequence of both direct and indirect effect on DNA. Direct effect of UV on DNA is two types: pyrimidine dimers and pyrimidine hydrates. So it's making pyrimidine dimers, that is, your pyrimidines are making a dimer between themselves, or sometimes pyrimidines are making hydrates. So that's how it's affecting the its effects of it. So ionizing radiations are also called uh, because they are ionization in the producing the ionization in the atoms. So they have two types: particulate and non-particulate. Particulate radiations are consisting of high energy atoms in particles generated due to the radioactive decay, and non-particulate ionizing radiations are represented by the X-rays and gamma rays, which are high energy radiations composed of photons. The genetic effect of radiations may be direct or indirect, and the direct effects of radiation is produced due to the ionization directly in the DNA molecule, while the indirect effect is produced through the ionization in the molecules other than the DNA molecule, and is believed to be mediated by free radical formation. So, what are the sources of radiations? There could be natural sources of radiations produced, so-called background radiations. These include cosmic rays from the sun and outer space, radioactive elements in the soil, terrestrial products. All these could be related to the geographical regions also. So, medical testing, nuclear testing, power plants, all various products like TVs, smoke detectors, X-rays, they are producing your uh, radiations. This was about physical mutagens. So far, we have discussed. Now comes the chemical mutagens. The first report of a mutagenic action of chemical was governed by Charlotte Auerbach, who showed nitrogen mustard to be the used in the poisonous mustard gas in the World War One and Two, and could be cause of mutation in cells. Since that time, many other mutagenic chemicals have been identified, and there's a huge industry is available for the food additives and industrial waste. So the mutagens, chemical mutagens, is first is the base analogs. So these are the chemically structurally resembles they resembles purines and pyrimidines and may be incorporated into the DNA. So by having the same shape or same structure, they could be incorporated into the DNA in the place of the normal bases during your DNA. For example, bromouracil, aminopurines are the two examples. So compound extensively used in research resembles thymine because it has a bromine in the 5 position instead of the methyl group. So 5-BU behaves like thymine and usually pairs with the adenine. So instead of T, you can have 5-BU also. Aminopurines are the ones adenine analog which can pair with the T or with the C. So causing A to T, G to C or G to C, A to C transition. Base analog cause all transition as do spontaneous tautomerization events are happening. So like A is T is wild type, then there's replications happening, then instead of T we have 5-BU is incorporated into DNA, then replication is again is happening, then you have AT and G. Instead of C we have 5-BU here also, and then in the replications also again you have G5-BU and transition mutations you have GC also. So like this 5-BU could be incorporated instead of another pair of your nitrogenous base pair. So chemical which alter the structure and pairing of properties of the bases. So they are mainly two examples: nitrous acids and nitrosoguanidine and methane sulfonate, ethyl methyl sulfonate, which reacts with the bases and add methyl to methyl groups. And third ones are the intercalating agents which get attached inside within the base pair. So acridine orange, proflavin, ethidium bromide used in the labs as dyes could act as a mutagens. So all are flat multiple ring molecules which interact with the bases of DNA and insert between them, and this insertion causes stretching of the DNA. DNA is stretched because of that and duplicates. DNA polymerase is full inserting extra bases and result in the intercalating agents cause a frame shift. So a frame shift is happening in this case. Uh, in the case of intercalating agents. So here's examples of proflavin as intercalating agents. In case like acridine, it just came and got attached here in between. Intercalated between the strands of DNA causes deletions or duplications of your additions of your DNA. So that's how the intercalating agents are working out. So there's agents who are also changing the DNA structure. So so this is called as catch-all category. So which includes variety of different kinds of agent. They include large molecules which bind to the DNA base pairs and cause them to be non-coding. We call them bulky lesions. Example is NAF. Agents causing intra and inter-strand crosslinks found in some vegetables and used in the treatment of some skin conditions, and chemicals causing DNA strand breaks, peroxides. So these are all the agents causing DNA structures. So how you can detect this mutation? For detection, you can do is calculate the number of colonies on the selective medium which are mutated and total number of colonies on the non-selective medium, and then multiply by 100. You can calculate the frequency of mutant cells in that case. So there are two techniques, a CIB technique. So this I will discuss at the end of your lecture. So that's it for today, students. Uh, that was small part of mutations at the end. It's because your CIB technique is somewhere in here. I have to teach you. So this will be flow with this then. So we do this part later. So for today we are done. So uh, I will share with you later the today's lecture. So thank you very much for today. I think we are done for today. So we are done with the cytoplasmic inheritance, mutations, We have studied quite in details about mutations today, various types of it, and how it is done. Yeah. So, any questions so far? How's your review is going on? How's assignments? I'm getting your assignments every day. That's really great. Okay, students, if you have no further questions, everything is good. Then we see us tomorrow, same time. So tomorrow I will be dealing with you day 4, right? So we will do some practical part. So we deal with the chi-square method, chromosomal abbreviations by chemicals in plants, induction of polyploidy, mitosis. So we try to do many practicals tomorrow as much as we can. So something about practical part also. So we cannot do them via our hands, but it's good to see the theory about it so that at least we know what we are gonna do tomorrow. So that's it for today. Thank you very much and see you tomorrow. Good night. Take care. Stay safe. Bye bye.